Thank you. Good afternoon, everyone. Today I'm going to talk about how we can do DNA computing by connecting it to stochastic computing. So the outline of this talk is that I first start by introducing stochastic computing, which is using electronic circuits for performing computation. And then I explain how we can do DNA computing using stochastic-based circuits. First, I talk about multiplier, which is a simple computation, and then I go for more complex computations like mathematical functions, and also I introduce our software that we developed for this type of computation, which is called fun DNA or function DNA. And finally, I talk about the future direction of this project. So we know that in positional binary encoding, we have bits that based on the position of each bit, there is a weight for that, and we just sum the weighted values of bits to achieve the value of the number that we are representing. Positional representation is very compact. By n bit, you can represent 2 to the n different values. But the problem is that performing operations, arithmetic operations by this representation needs complex circuits. For example, for multiplication of two numbers A and B, if we want to do computation in binary, positional binary, we need to first calculate the partial products of the numbers, and then we add them together. So for example, this is a 3-bit multiplier that you see it has some gates, AND gates, and half adder, full adder, and so to perform this 3-bit multiplication. If we consider each half adder has 2 basic gates and each full adder 5 basic gates. In total, we need 30 different gates to perform a 3-bit multiplication. The question is that, is there a better or a lower cost method for performing computation? And this is where we have stochastic computation. I introduced the idea by a simple example. Assume that we have 2 boxes and each of them has 10 black balls and 10 white balls. So what is the probability that we take one ball from the first box blindly and another ball from the second ball, and both of them are black? So it will be 1/2 taking one black ball from this and 1/2 from that. And the total probability of having 2 black balls will be 1/4, which is the multiplication of half by half. So you see that if we work with probabilities, we can perform computations in this method, multiplication in this method. So in stochastic computing, the idea is like that. We are presenting or representing numbers by random bit streams of 1s and 0s. And the value of the number that we are representing is proportional to the number of 1s that we have in the bit streams. If we have more 1s, it means that the number that we are representing has a bigger value, and if we have less, it has less values. For example, here, for representing 3/7, we can have 7 bits, and 3 of them are 1. Of course, because this is not positional representation, we can have different representation for this. And these are all representing the same number. So based on that, we can actually perform multiplication in this method by a simple AND gate. If we represent our numbers by random bit streams and we feed them to the input AND, the inputs of the AND, and get the output will be the multiplication of these two because the inputs are random. I should mention that we assume that bitstreams here are independent. So for this example, for example, you see that 6/8 is first number A, and 4/8 is the other number. And the result is 3/8, which is the multiplications of these two numbers. So in this way, we can perform, if we compare it to the binary that we needed 30 gates, we can perform multiplication by a simple AND gate. We can actually have other arithmetic operations. For example, we can calculate 1 minus multiplication by using a NAND gate rather than an AND gate, which I guess is obvious because we just flip the outputs and the probability will be 1 minus what we had before. And a multiplexer here can perform a scaled addition. It means that the output will be the scaled addition of the two inputs that we have. So now I'm going to talk about DNA computing. I guess you know that DNA computing means that we perform computation by DNA molecules. And there are different types of computations, but for concentration-based DNA computing, we can represent the numbers that we have based on the concentration of molecules, actually DNA molecules. For example, and we can have different types of representations. For example, we can have direct encoding, which we use only one molecular type, and the value of the variable that we want to represent is proportional to the concentration of that molecular type. Another type of representation is dual rail. In dual rail, we use a pair of molecular types, and the value that we want to represent is the difference between these two molecular types that we use. The benefit here is that we can show also negative numbers. For the first one, we couldn't. And the third one that I introduced here is fractional encoding. Again, here we use a pair of molecular types, let's say A1 and A0, and the value that we want to represent, A here, is the concentration of one of them, A1, divided by the total concentration. What's the benefit of this? Here, if we use fractional encoding, we can make a bridge, a connection between stochastic computing and DNA computing. So assume here each bit of the random bit stream that we had is represented by one molecule. So for example, each 1 is represented by A1 and each 0 is represented by A0. So in this way, we can represent our numbers by molecules. The question is that how we can implement an AND gate to perform multiplication. So we need to design a set of chemical reaction or a set of reactions such that it mimics or implements the behavior of an AND gate. And these are the 4 reactions that we can use in order to implement an AND gate. Why it is working like an AND gate? The reason is that the output C1 is achieved only when we have both inputs A and B are chosen from A1. So when A1 and B1 react together, we will have C1. Otherwise, always we will have C0. The same way that an AND gate works. And also we can implement a NAND gate similarly by flipping the C0 and C1, so the products that we have. And these 4 reactions are implementing a NAND gate by molecular reactions. And the result is 1 minus the multiplication of 2 inputs that we have in fractional encoding. So based on this connection, we proposed a systematic method for performing computations using a stochastic DNA computation, using a stochastic computation for mathematical functions. So our method is first we have the function that we want to implement. We we obtain the series expansion of that, like Taylor series or Maclaurin or whatever series expansion that we want. And then we map this expansion to a cascade of AND gate and NAND gates that we introduced earlier in molecular implementation. And then we write the chemical reaction for each gate that we have, and finally, we map the chemical reactions to DNA reactions. So going from abstract mathematical representation of function to its DNA oligos. So the way that we map chemical reactions to DNA reactions is based on this paper, and in that method, each molecule is represented by a single strand DNA that has 4 segments. I call them domain 1, toll 1, domain 1, toll 2, D1, T1, D2, T2. And based on that, I can implement all the reactions that we had. If you have noticed, we have all the reactions that have 2 reactants, 2 inputs, and 1 product. So it means that we need to implement this type of chemical reactions by DNA reactions. And again, based on this paper, we can design those segments of single-strand DNA to perform the computation in that way. So here we have X1 and X2 are 2 molecular inputs, and the results here will be X3. So this sequence of 3 DNA reactions can implement this 1 molecular or chemical reaction for us. So every reaction that we have, we map them to this set of DNA reactions and optimize them. I'm not going through the details of that, but this is the way that we map these chemical reactions to DNA. So here I have one example of how we map the the functions to chemical reaction. Let's assume that we have exponential function. Then we obtain the Maclaurin series of the function here. We truncate it at some point based on the accuracy that we target. And then we reformat it. For example, here we have used Horner's rule. And excuse me. In this representation, you see that each of the parentheses that we have has the form of 1 minus multiplication of 2 numbers. For example, here 1 minus x multiplied by 1 over 5. And when we go to outer parentheses, we have this for all of them. And then we can map this equation to this set of cascaded NAND and AND gates. So this is implementing this equation. And finally, we just write the chemical reactions for each gate, and then we map them to DNA reactions. We have actually designed this set of gates for other functions, like basic functions that you see. And also, we have developed a software to do this automatically for us. So in the software that we have, here is not the good quality, it's blurred a little bit, but we choose the function that we want, and also the input that we want to calculate the function. And then after we press this save button here, the representation of function, the series expansion, is calculated by the software. The chemical reactions that we need are generated here. And finally, the oligos or the nucleotides that we want to implement these chemical reactions are generated here automatically. We use other works for this part of the transfer between chemical reactions to oligos. But the software in general was developed by us. The future direction for this project is to implement DNA artificial intelligence systems, and we have actually already have the basic units that we need. For example, for a simple perceptron, we have the multiplication to multiply each input by its weights. And also we have implemented this sigmoid function. So we can implement this, and we are going to implement more complex circuits by cascading or connecting perceptrons together. The preliminary results of our simulation using MATLAB, which are shown here in the blue. I don't know if you can see the difference. And what we have for the simulation of DNA reactions are matching. It means that if we consider the perceptron that we had here is a binary perceptron that has either true or false in the output. And here we use a threshold, for example, 0.5. Always the MATLAB simulation for the perceptron that we have and the output of the perceptron that we implemented by our reactions are matching. Means that they are always either more than half or less than half. So finally, I want to acknowledge the team that are working on this. We have a postdoc who is doing the experiments for this project, and also other students who are working for the software development and other parts of the project. The project is supported by National Science Foundation. Yeah, I guess that's it. If there is any question, please go ahead. Thank you.
Questions?
Thank you for your talk. I was wondering if you had plans or if you had done in the past any experimental work.
Yeah, that's a question I was expecting. Thank you for asking. Actually, we have already validated the basic logic gates that we introduced here, like the AND gate and NAND gate. We have experimentally implemented them in the lab, and we got the results that validated. But we are planning for having more complex circuits, like having the output of this single AND to another AND, and so extending it. So we have not experimentally validated the whole circuits, but the basic units are already validated experimentally. And we are planning to, again, validate other parts and submit a paper for that.
Thank you, Ahmad. That was a great talk. I really enjoyed your work. Of course, I didn't understand many parts. This was very mathematic.
Maybe that is why you enjoyed it.
Yeah. My question is about, for example, can you go to the slide 17 or 18, I guess, for some calculations?
You mean mapping?
The one that you did some calculations together for a function. Do you have an idea how long does it take to get the result from the beginning?
Yeah, that's a good question. Unfortunately, in general, inherently chemical reactions and DNA reactions are slow. Based on our experimental results, it takes maybe I have something in the hidden slides, around 3 to 4 hours for each sample to get the output or collect the output for each thing. And obviously, when we increase the accuracy, it means that we are adding more terms here, and we are adding more gates here, and the latency of the system is increased. So if we want more accuracy, we have a longer circuit, and the timing also is extended. It means that we need more time to collect the output for each input sample. But now, currently, for this example that I mentioned, 3 to 4 hours is a good time to get or collect the outputs. Did I answer your question?
Yeah. Okay, thank you.
Other questions? I have a question.
Please.
Your software open source and is it available?
Yes, we have it in the GitHub. I forgot to include it. But if you go to the Cut Lab at UKY, University of Kentucky, you see that the software is open source and it is available on GitHub to use it. And please, if you use it, you see any bug or any error, get back to us with your feedback because we are still improving our software.
Anyone else? Okay, thank you.
Thank you.